Howdy! So when we last chatted, we talked about duplicated chromosomes. So chromosomes are able to duplicate, so DNA copies itself. We had a whole lecture on that already. So you go from one strand of DNA to two, but when they're first produced, they're connected a bit. And remember, if we zoomed in on this DNA, it would be wrapped up, really tight. That's why we can see it with a microscope. That's when it forms a true chromosome, when it's visible under a microscope. So let's put some lingo on this. So here is a chromosome consisting of a single chromatid, meaning it's just one stick of DNA wrapped up. So if we were to zoom in on this, we would see DNA wrapped up around proteins. So here it is. Now, DNA duplicates, and when it does so, it's stuck to a clone of itself. So it goes from this to this, okay? And this is a single chromatid. So there's several words here, chromatid and chromatids. Let me try to write this all out. Let's see here. A chromosome is composed of chromatids, which are basically like sticks of DNA and protein, and chromatids are composed of chromatin, which is DNA and proteins. Okay. All right, so here we have the duplicated chromosomes. So we have these two chromatids. They're known as sister. Think of them as twin sisters, sister chromatids. And this is still considered one chromosome. Students run into so much trouble because they forget that this is one chromosome. So if you see this and I ask you how many chromosomes is in this cell, this would count as one chromosome. Okay, but later on during mitosis, after mitosis, these would separate into individual chromosomes known as daughter chromosomes. They go from being sister chromosomes to daughter chromosomes, okay? And that is the basic process that's going on in this chapter. And this is the lingo. You should be able to draw this and label this well enough to know it. So ultimately what's going to happen here is we're going to have a cell with a certain number of chromosomes, here represented by 1. So here's a chromosome composed of a single chromatid. It's really a single double helix of DNA, all wrapped up really tight so it's visible. It's going to replicate, which we talked about in a previous lecture. So that's really 2 strands of DNA now. And then during mitosis, these two are going to separate out into different cells. And that is how the cell is going to reproduce and send equal copies of DNA to both sides. Mitosis is really making clones. We're making exact copies. So notice we start with this and we end up with 2 of them down here. So we've made copies, and that's what this whole process is about. In your cells, as we've said already, this is, by the way, this is known as a karyotype. That's I don't know why it does that, but that's an image of all of your chromosomes at once here. So here's an individual that's female. There's 2 X chromosomes down here. X chromosomes are the same size, whereas a Y chromosome would be really small. And so we have all 23 pair of chromosomes. So this is a diploid individual. They're all shown here. Now we're going to have to move those chromosomes all around in a cell and get the right number of copies into one cell and the right number of copies into another cell. All right, so there are some important parts in this process of mitosis that we need to understand, some cell parts. So first of all, a definition: mitosis is a type of nuclear division, so division of the nucleus, that produces identical nuclei. It's making clones. Cytokinesis is the fancy word for the division of the cytoplasm, which forms 2 new cells. So mitosis is really just division of the nucleus. So a couple of important parts. The spindle, also known as the mitotic spindle. This is a cytoskeletal structure. So if you want to review that, go to the cell chapter earlier on in the book. But the cytoskeleton is composed of proteins. It helps hold the cell together. It helps to move things around. So it's a cytoskeletal structure that pulls chromatids apart It's made up of a type of structure known as a microtubule. If you zoom in on those parts, they're little thin tubes. Then there are structures known as the centrosome. And actually, it's more of an area than a structure. So the centrosome is a microtubule organizing area or organizing center. And in animals, it has two centrioles and an array of microtubules called an aster. So if I was going to draw that, here's a cell. And the centrosome is just an area here and an area here, let's say. And in an animal, you might have an aster, which looks like an asterisk, which just means several microtubules overlapping. And then the centromere, the centrioles look like little trash cans laying down here, and you don't have those in plants, which is a good test question. Do plants have centrioles? Plants do not have centrioles. The other thing that is going to be a little tricky in this chapter, as you heard me misuse a word there, is there's a lot of words in this chapter that start with centro or centri. Centrioles, centromeres, centrosomes, okay? So centrosomes is this area right here. That's the microtubule organizing area. Okay, and if you forget what a centromere is, that's that area in an individual chromosome where they're kind of stuck together. And I haven't drawn any of those in this cell. This is just the microtubule organizing centers of the cell. So here's a picture of a cell. Now I could ask you on a test, is this a plant cell or an animal cell? You would say, well, that's a plant cell, Professor Karyotype. That's easy. There are no centrioles down here. I didn't draw any of those little trash candy looking things because plants don't have those. But you can see very clearly the microtubules. Those are the part of the mitotic spindle that connects to the chromosomes. And you can see the centrosome, that area where they're organized. You can see the chromosomes quite clearly there. Okay, so the last thing I want to talk about in this video before we proceed to the stages of mitosis is the basic cell cycle. And you can see that cycle here. This is the life of a cell. And it's composed primarily of an overall phase known as interphase. Interphase is the bulk of a cell's life. And it's really broken down, if a cell is actively growing and dividing, it's broken down into three parts. G1, or also known as GAP1, S, and G2. So G1 and G2 phases are gap phases, but you can think of them as phases where the cell grows and gets ready to divide, or well, basically where it's growing. It may be making copies of organelles like mitochondria and things, but it's getting ready to divide. M phase, which is this one right here, is mitosis or mitotic phase. That's what this chapter is mostly about, but it's only a very short period of time in the cell's life. So your book says if an animal cell divides once a day, that's probably not likely, but let's say it does. For most of your cells, that's not likely anyway. Mitosis would only be about an hour or so. Now, S phase is when DNA copies itself. It stands for synthesis phase. Hold on a minute, I'm having trouble here. Synthesis phase, that's when DNA replicates. We had a whole chapter on that. That's when helicase unzips the DNA and all that stuff happens, okay? So a cell, let's just look at what happens here. A cell recovers from dividing from mitosis. It's small. Now it gets bigger, right? The cell grows. Then DNA gets copied. Then the cell probably grows some more and gets ready. And then it divides by mitosis. So that's the basic life cycle of a cell. Now, not all cells are always dividing. Some of them just sort of hang out in essentially G1 phase after they get big enough. But if a cell is actively dividing, this is the basic life cycle of that cell. So in the next video, we'll talk about how cells divide. But let's just look, before we go, at a summary then of the cell cycle. In G1 phase, or gap phase 1, that's the longest phase of the cell cycle. That's where cells grow. They were just born by mitosis and then they grow. S phase, they replicate DNA. Gap phase 2, organelles replicate. So you might make extra copies of organelles to get ready to divide. Microtubules might begin to organize to get ready to divide. The cytoskeleton prepares. That's collectively, all of that together is known as interphase. Mitosis is divided then into 5 phases. So M phase is divided into 5 phases. And we'll talk about those in the next video. After M phase, or at the end of M phase, cytokinesis kicks in, and we divide into 2 new cells, and it's often abbreviated as C, as you can see there. Now, I mentioned that a lot of cells kind of grow in G1 phase and don't divide. Those cells are said to go into G0 phase then, meaning they're not going through S phase and G2 phase. They grow up big and now they're doing their job and they're just hanging out. And so that's the terminology they use for that. Instead of G1, G2, they go into G1 and then they just sort of go into a holding pattern. They hang out and do their job, and that's called G0. All right, so in the next video, we're going to start then with the actual phases of mitosis, and we're going to draw those, and we're going to talk about what's happening in the nucleus and in the rest of the cell during that process.